Hello, this is Liskin Swint-Kruse from the Department of Biochemistry and Molecular Biology at the University of Kansas Medical Center. I'd like to share a story with you about rheostats and toggle switches for modulating protein function. This is an interdisciplinary story with roots and motivation arising from developments in genetics. We're now in the era of $1,000 genomics, and we can cheaply sequence the genomes and exomes, that's the protein coding regions, for hundreds and thousands and hundreds of thousands of both patients and organisms. And our rationale is that this will enable us to match patient cohorts with the best drugs, diagnose genetic disease, tailor therapies to cancer genomes, and identify predisposing risk factors. But to accomplish this, we must first solve what's been called the million-dollar interpretation problem. This arises from a number of scientific limitations, and I've focused on the last one on the list, the fact that there are huge numbers of differences among individuals. In fact, the exomes of non-relatives can differ by more than 10,000 amino acids. Many of these will not have medical consequences, so it's a needle in a haystack problem to figure out which ones matter. What's more, these differences are too numerous to assess in the lab, so we have to have computational methods to identify them. And indeed, dozens of computational algorithms have been created in the past 20 years, but their success rates need to be greatly improved to be of use in the clinic. We decided that, rather than attempt to devise a new algorithm, we would examine the input and the assumptions that underlie existing algorithms. And in doing so, we realized that they all have a set of commonalities. First, as input, they all use evolutionary information in the form of a protein sequence alignment, and this is treated as nature's mutation experiment. If an amino acid is present in a column, it's considered to be good, and if it's absent in a column, it's considered to have been bad for the protein and eliminated by evolutionary processes. There's also a set of common assumptions that underlie these algorithms, and in fact, we're now teaching these to freshman biology students. And so what we teach them is this. At important positions in the protein, if we make a change, we expect most of the amino acid substitutions would damage either the structure or the function of the protein. A few amino acids might allow normal or near normal function, and we can guess what these would be because they would have similar physical and chemical properties, like serine and threonine both have a side chain hydroxyl. We would also see that these were the amino acids present in the sequence alignment during evolution. The last assumption comes because we are using a sequence alignment, and that is, we're making the assumption that if we know something about an amino acid change at one position in one homolog, we can extrapolate that and know what the outcome will be of that amino acid change in any other homolog. In thinking about this common input and textbook assumptions, we realize there's a disconnect because these substitution rules were based on experiments that were biased towards conserved positions. Those are the ones that do not change much during evolution. But if you look at a sequence alignment for most protein families, many of the positions, many times more than 50% of the positions, do change during evolution. They are not conserved. Furthermore, we know that a lot of these positions are important for differentiating and evolving variation of protein function. So in this particular example, here are 3 venous flytrap fold proteins. Each binds a small molecule in this central cleft. But the chemistry of these molecules are very different, and the amino acid side chains that interact with them must also be different. So non-conserved positions can be important to protein function. So we ask the question, do these textbook rules apply to changes at non-conserved positions? And we were particularly interested in this third rule, that we could extrapolate the substitution outcome from one homolog to any other homolog. And we wanted to test it using multiple homologs in the same family. And so we thought of an experimental design which essentially recapitulates a sequence alignment in the lab. We needed to have multiple homologs on which we could do experiments. We wanted to look at the non-conserved positions and make changes at those positions. We wanted to put multiple amino acids at each position, and we needed a quantitative readout that was directly related to functional change. So this design has several technical and interpretation hurdles. We were able to overcome these limitations by using the family of Lacl transcription regulators. Many of you may be familiar with the founding member of this family, the E. coli lactose repressor protein, but it's just one protein of a very large protein family. We used 10 paralogs in this family that we modified to bind the same DNA sequence, and then we made mutations in analogous positions for all 10 of these homologs. We then assessed the mutation outcomes using a high-throughput assay based on their common function of transcription repression. So these proteins have two functional modes. They have a repression mode, where there's high affinity for a very specific DNA binding site called the operator. And when the repressors are bound to the operator, RNA transcription of downstream genes is not allowed. Then there's also an allosteric induction mode, in which there's some small molecule effector, usually a metabolite, that binds to a different site on the protein, causes some sort of protein conformational change so that the repressor no longer sees the operator as a special region of DNA. The genomic DNA competes the repressor off of the operator, and that allows the transcription machinery to transcribe the downstream genes. Many of you have already seen such assays if you've ever carried out a blue-white screen. Here's an example of wild-type Lacl repressor. In its repression mode, it's repressing transcription of downstream beta-galactosidase gene, and so colonies are white. But if we add the small molecule inducer and give the beta-galactosidase a colorimetric substrate, the colonies become blue. On the right are an example of transcription repressors with point mutations, and you can see that some are very strong repressors and make white colonies. Some are modest repressors and make light blue colonies, and others are poor repressors and make darker blue colonies. Now, we mentioned that we wanted this to be quantitative, and these plate assays aren't quantitative, but there is a version of this assay that can be done in liquid culture and assayed on a spectrophotometer. And this is an example of that assay where the white colonies now correspond to clear solutions, and the blue colonies correspond to dark yellow wells. And then when we quantify this with the spectrophotometer, we get bars that look like this. The low bars now are good repressors, and high bars are poor repressors. And this is quantitative over 5 orders of magnitude. Furthermore, by doing it in plates, we could do it in a medium throughput method that allowed us to assess around 1,100 variants. We had control experiments in place that let us show that all of the proteins are expressed and capable of binding DNA, even if the DNA binding wasn't strong enough to allow transcription repression. We had several other series of control studies in which we purified series of these repressors and directly measured their DNA binding affinities, and we could show that the change in repression correlated very tightly. We could also do in vivo growth assays and show that the changes in repression were large enough to alter the bacterial growth. So with all those control experiments in place, we now took each parent protein and we made multiple mutations at each of the non-conserved sites and assayed their transcription repression. So when we take all the data for one position in one protein, what we expected to see most of the time was a plot like this, where here's the transcription repression for the parent protein, both in the absence and presence of inducer. Here are 2 substitutions at position 50 that are about like wild type, and then all the rest of the substitutions at position 50 kill the ability of the protein to repress. So they're like the control with no repression present. And this is what the textbook rules would predict we would see for most positions. And we've started to think of this as a functional toggle switch. The protein's function is either on or it's off. However, we didn't see this very often. Instead, most of the time we saw a very different type of pattern that we call a rheostat pattern. So in this case, here's our starting protein with the striped bars, and some mutations make repression worse and some mutations make it better. On the right, here's a second example with the striped bar for the starting protein, and some mutations make it worse and some mutations make repression better. And this rheostat pattern dominates our data set. We had 135 positions that we assayed spread out over 10 homologs, and 101 of them had the rheostat pattern. Only 10 of them had the toggle pattern. 18 had kind of complicated patterns that we couldn't assign to any particularly category. And only 6 were neutral. That is, it didn't matter what you put there, the protein function remained the same. The second surprise was when we looked at side chain chemistries. So if we look at these in the rank order from best to worst and try to find out, well, what drives these changes? If we look at hydrophobicity, if hydrophobicity were driving it, we should see a continuum of color from green to white to magenta. But you see here that clearly hydrophobicity doesn't correlate with what's the best repressor. And in fact, when we look at the same data set, color-coded by lots of different physical scales, no patterns emerge. And with this publication, we have an 87-page supplement showing that there are no physical chemical trends among the side chains that were involved with these rheostat positions. We then got to our original question, how well do outcomes compare among homologs? And again, we see no common trends. So in this case, what we did was we took the repression and we normalized it so that the parent chimera repression was 1. And then if the mutation made repression worse, it dropped below the line. If it made repression better, it came above the line. And so you can see here at position 51, if you had a glycine, there were cases where glycine was a neutral substitution. There were cases where glycine was better than wild type, and there were cases where glycine was catastrophic. What's more, there were no correlations with evolutionary propensity at this position. So glycine is actually not very represented in the data set. Histidine's not represented at all, but here's a histidine that's enhancing, and there's several glycines that are enhancing. And on the flip side, leucine is highly represented in this data set, but there are leucines that are detrimental. So if we go back to our question, do the rules apply to non-conserved positions? The answer for all of these rules was no, they don't. And that got us thinking, if rheostat positions don't follow these canonical substitution rules, these must be the positions where computer algorithms are failing to make good predictions. So we addressed that explicitly. We separated out a set of toggle positions in wild-type Lacl repressor and a set of rheostat positions, and we worked with Dr. Jana Bromberg at Rutgers University and Max Miller to do separate predictions for each of these types of positions using 16 different algorithms. And what we found was that generally substitutions at toggle positions were correctly predicted to be deleterious, but the predictions of outcomes at rheostat positions were very poor. By now, you may be wondering whether this is just a special region of a special protein family, but that does not seem to be the case. So there's a data set available for wild-type Lacl from Jeffrey Miller's lab, in which they made about 4,000 substitutions at 329 positions across the whole protein. These were done in the 70s and 80s, so the assays are slightly lower resolution, but we can examine them and see that across all of Lacl repressor, about 40% of positions are rheostats. Only 12% are toggle positions. We've also been able to see that this is not a special protein family. So we've worked with the Fenton lab at KU Med to show at least 2 globular soluble proteins have rheostat positions. And we worked with the Hagenbuch lab to show the existence of rheostat positions in membrane transport proteins. And there are many other data sets that have come out in the past few years, which when we examine, we find that there are rheostat positions. We're not the first people to show that amino acid similarity rules don't always hold. Several other papers have noted this. We're also not the first to show that evolutionary amino acid frequency is not predictive of outcome. several other papers have shown and noted this. So in summary, I hope I've convinced you that rheostat positions are widespread within proteins and among proteins, and that they do not follow the canonical substitution rules. At this point, I'd like to give you a little preview about what might give rise to these non-canonical substitution outcomes. Up till now, you might be thinking, well, please show me where these are on the structure. Here's a structure of Lacl repressor. The functional unit of all of these family members is a homodimer. So one homodimer binds one DNA operator. One monomer here is in magenta and one is in purple. And then in white is the region that we focused on for this first mutagenesis study. And I've blown it up here on the right. The green positions are highly conserved. The magenta ones are moderately conserved, and the white positions are not conserved among the homologs of the Lacl family. And we mutated all of the white side chains and many of the magenta ones in the study that identified rheostat positions. When we think about these on the structure, we can't explain rheostat outcomes by the proximity to DNA. Some of them directly contact DNA and some of them do not. We cannot correlate it with how buried or exposed any position side chain is. There are, though, in this region, local side chain repacking when we compare different paralog structures. And we also know that this is a region where dynamics are very important and that there are dynamic changes that occur with DNA binding and that there are dynamic changes among the paralogs. It's beyond the scope of this current talk, but in collaboration with Dr. Oskin and Paul Campitelli at Arizona State University, we have described very compelling dynamics changes that are associated with substitutions at rheostat positions. The other ongoing questions that we're addressing is how non-conserved are rheostat positions, which types of proteins contain rheostat positions, and where are they located in each protein. There are several take-homes for life scientists. First, know that the current prediction algorithms about mutation outcomes do not work for rheostat positions. We find it very helpful to change our frame of reference. We think about what the position does rather than what the amino acid residue does. Anytime that you identify a substitution with an intermediate outcome, that's very likely to be a rheostat position, and it won't follow canonical substitution rules. If you want to identify whether a position acts as a rheostat toggle or neutral position, what you need to do is generate 10 to 12 randomly chosen amino acids for each position. The other take-home realization from this is that disease mutation datasets and alanine scanning, each of which only have one or a few amino acid changes per position, are incomplete and won't show you the whole picture about that position's role in the protein function. And finally, I showed some examples, and it seems to be consistently so, that some variants, somewhere around 10%, are better than wild type in all of the studies that we've carried out to date. I want to acknowledge and thank all of the Swint-Kruse lab members involved in this work, as well as the NIH and the WM Keck Foundation for funding prior and ongoing studies by our team of collaborators. In the work shown for this talk, Susan Egan provided the protocol for quantitative beta-galactosidase assays, and I described some of the results from our collaboration with Jana Bromberg. I also showed a small fraction of the data that had been generated by collaborations with Drs. Aaron Fenton and Bruno Hagenbuch at KU Medical Center. In the course of this work, we generated several computer programs for which the code is available, and the links to these and several other resources can be found at my website. Thank you for listening, and I hope you'll examine your own favorite protein for the presence of rheostat positions.